let's talk just a little bit about DNA. Now, we will talk a ton, or I will talk a ton about the structure of DNA later on in the course. But we should just introduce some important concepts of DNA to start. So I will often diagram DNA like this. So 2 lines, and then I'll do something like this. So these little boxes, these are going to represent bases of nucleotides. So DNA is made up of nucleotides, well, 2 strands of nucleotides in double-stranded DNA. Now, DNA can be single-stranded under certain situations, but unless I specify single-stranded, we will always assume DNA is double-stranded. And here we have our 2 strands, and the 2 strands are held together by hydrogen bonding between the bases of these nucleotides. So I will often diagram DNA something like that. I'll make diagrams like that, and sometimes I won't even show the bases. I'll just do something like this. And DNA is said to be a double helix. So this twists, but obviously I'm often not going to diagram the twist in the DNA diagram. Now these things right here, we're going to call these phosphodiester backbones. We'll learn more about that. And just briefly, so what is a nucleotide? Well, we are going to learn a lot about the structure of nucleotides, but a nucleotide is made up of a phosphate group, a 5-carbon sugar that looks something like this, and there's an oxygen in that 5-carbon sugar, and a hydroxyl group down there. And this carbon right here, this carbon atom, is attached to the nucleobase. And these are the nucleobases up here. So again, we don't need to focus on the structure of the nucleotide at this part of the course, but I did want to bring something up because it's often very confusing for students. Now, I'll probably talk, it's hard to not talk about DNA base pairs throughout the genetics course. And the 4 bases of DNA, nucleobases of DNA, are A, C, G, and T, right? So adenine, cytosine, guanine, and thymine. Now, sometimes you're going to hear terms like this: adenosine, cytidine, guanosine, and thymidine. Now, these are very similar. These are very similar sounding words, right? Well, it turns out, so these are the names of the nucleotides. And these are the names of the nucleobases. Now, I won't expect you to remember which is which, but I just am using this opportunity, the first lecture, to help you see that, when you hear these 2 things, it's not that, you don't know what the 4 nucleobases in DNA are. It's just that these names, they're very similar and they're hard to keep track of. And I may sometimes, when I mean to say adenine, especially if I'm talking quickly and thinking quickly, I may substitute adenosine in there. So I just want you to know that these terms over here, the terms for nucleotides, are very similar to the terms for nucleobases. And I won't expect you to know which is which. I won't test you on that. But it helps to understand that there are two similar names for these related things, for each of these related things. Now, we know the nucleus contains DNA, but in the nucleus, DNA is not naked. It exists, it is found, I should say, in chromatin. Chromatin. So what is chromatin? So chromatin is a complex of DNA and proteins. So let's say this is a strand of DNA. So unless I say it's single-stranded, always assume DNA is double-stranded. So that's a double-stranded DNA strand. Let's say in a nucleus. So if I say nucleus, we know we're talking about a eukaryotic cell because prokaryotes don't have nuclei. Now, in the nucleus, DNA is going to be found in a complex with lots of different proteins, but most commonly histone proteins. And I have just diagrammed an octamer of histones. So histones are proteins that help package the DNA and organize the DNA. And there are 4 major types of histones. There are more than that, but four common histones that get together, 2 of each type, that form this octamer of histones. And the DNA wraps around, binds to the histones and wraps around in a specific order. I think it wraps around 1.7 times or 1.67 times. And this complex of DNA and histones here is often called a nucleosome. So chromatin essentially is DNA and proteins, but the DNA is found in nucleosomes, in chromatin. So chromatin, again, is a complex of DNA and proteins. And if we are examining chromatin, we should be able to see nucleosomes. Now, if that's one nucleosome on the left, and here is another one, each of these histones is being represented by a circle, and I'm showing this octamer of histones here, and I'm going to wrap around that DNA again. So we have a second nucleosome over here, and that DNA between the 2 nucleosomes is called linker DNA. Now, you can see this DNA, this linker DNA, well, it's relatively long, right? And when chromatin looks like this, we can say this is open chromatin. So in open chromatin, the linker DNA, the length of the linker DNA between the nucleosomes is relatively long. And that's a good thing because it means, well, sometimes it's a good thing when there are proteins that need to access that DNA and read information from it. Well, the DNA should be in an open conformation in order for these proteins to get at that DNA and get the information. Now, chromatin can also be in a closed state. Now, when it's in a closed state, we think it looks something like this. You know, this is still really hard to do because even our most powerful microscopes have a difficult time looking or examining the structure of DNA inside the nucleus. So I should have diagrammed this one a little closer. I'm going to stick another nucleosome in here. So, and let me change the color for the DNA. So here, this very little linker DNA between the nucleosomes. So we're going to call this closed chromatin. And there are a couple of different terms that are used to refer to open chromatin and closed chromatin. So let's say we have open chromatin and closed chromatin. Sometimes the open is called euchromatin, and the closed is called heterochromatin. So open chromatin is often said to be decondensed, and heterochromatin is said to be condensed. Now, inside the nucleus, we could have a long, how about this? So DNA inside nuclei exists as chromatin, but the DNA is also organized into chromosomes. Chromosomes. So essentially, chromosomes are long DNA molecules. And chromosomes can be linear. They're linear, typically linear in eukaryotic cells. We will see that our mitochondria have chromosomes also, and those are circular. But in prokaryotes, they're typically circular. And so, okay, what do we mean by linear? Okay, so here's a double-stranded DNA. That's a linear double-stranded DNA, and our chromosomes in our nucleus are really long linear DNA molecules. And in prokaryotes, they're circular. There's no free ends. So here's a linear molecule there. There's an end here. There's an end there. There are no free ends in a circular DNA molecule or a circular chromosome. Now, during, let's say, most of the cell cycle, I'll talk more about the cell cycle in a moment. So the DNA of a chromosome can be in various stages of condensation. So you can have some parts that are closed, some parts that are open. And this is a really important area of research right now. So here's a nucleus, and let's say this is the DNA of one chromosome. I'm going to diagram it as in the decondensed open state. And maybe there are some more condensed regions of this chromosome, and I'm just kind of making a messy diagram here. So for most of the time, or I say most of the cell cycle, the DNA of the chromosomes, of most of the chromosomes, are thought to be in this decondensed state, but it's not disorganized. So how the DNA is organized in the cell is a really important area and really hot area of research right now. So nuclei are thought to have certain territories, and each chromosome is thought to have its own territory. That's really poorly understood, but what I want to show you is that, during most of the cell cycle, most chromosomes are open, they're decondensed. But during mitosis, well, the chromosome condenses. And it's thought that this is necessary to help mitosis, accurately place DNA in the two daughter nuclei that result from mitosis. So I'm often going to diagram chromosomes like this. So this is one chromosome in a cell, and it's condensed. And this thing right here would be the centromere. I'll talk more about that in a moment. These are the two arms, and this is a long linear DNA molecule. And if we could look down somewhere, we might be able to find the end over here. And these ends, this is called the telomere. And telomeres are thought to have a special structure that protects them from degradation. So, okay, there might be an end in here. There should be somewhere in there. So, and that would be the telomere. Now, when I'm diagramming a chromosome, I'm probably not going to diagram it like that. I'm going to diagram it in the condensed state, whether we're talking about mitosis or meiosis, or what we would call interphase, when the cell is not in mitosis or meiosis. Now, chromosomes, when scientists first started seeing chromosomes under a microscope, and they could really only see them during mitosis when they're condensed and when they're using a stain that binds to DNA, is they would see chromosomes with different structures. And we have a couple different names for chromosomes based on their structures. So this one over here, this is called a metacentric chromosome. This is an acrocentric, and this is a telocentric. So in a metacentric, okay, let me talk about this thing first. What is this? This is the centromere. So these names are based on the position of the centromere in the chromosomes. And the centromere was first defined as the constriction point. So there was a point of most chromosomes where the chromosome got skinnier. You know, scientists were looking at the chromosome under a microscope, and the chromosome was stained, and they saw a part of the chromosome that was skinnier than the rest. That constriction point is now called a centromere. And the centromere is not always found in the center of the chromosome. In a metacentric chromosome, it's approximately in the center of the chromosome. And as a result, we have these two arms. So here's one arm here and one arm here. And those arms are approximately equal sizes. So over here in an acrocentric chromosome, the centromere is closer to one end of the chromosome than the other. That results in one chromosome arm being shorter than the other. And so, and when there are two arms and one is clearly shorter than the other, the arms are named P and Q. P, this might be an urban legend, but P, I believe, comes from the name petite, the French word petite for small or short. So the P arm of a chromosome is supposed to be the shorter arm of that chromosome. Now in a metacentric chromosome, someone has to, just define one arm as P and one arm as Q based on, how that arm stains relative to the other one. So without other information, we can't know which arm is P and which is Q for any chromosome from a given organism. If they've already been defined, we're going to need some other information to know which one is P and which one is Q. But if there's a clear difference in the sizes, the shorter one is P, the longer one is Q. Now some chromosomes, I believe chromosomes in mice, are all telocentric chromosomes. And what that means is the centromere is found at one end of the chromosome. And there aren't two arms, there's only one arm, and that arm is Q. There are no short arms, it's said that there's only a long arm. We call that the Q arm. So telocentric chromosomes have the centromere at the end, at one end of the chromosome. So what will we talk about next? I lost my notes here. Okay, we'll go on to the next subject. I think it's going to be the cell cycle.